welcome back to the Igniting Curiosity in Life Science. So today we will dive into the cell's power generator, that is the mitochondria, and uncovering its electric secret, that is what the mitochondrial membrane potential. Okay, let's start the mitochondrial membrane potential. So what is mitochondrial membrane potential? So mitochondrial membrane potential is the difference in electrical charge, that is voltage, across the inner mitochondrial membrane. Okay, it is essential for ATP production, the main energy currency of the cell. So mitochondrial membrane potential, it is mainly denoted as the delta psi m. Okay, m is the mitochondrial. So that is what the representation of the, or the symbol of a mitochondrial membrane potential. So why it is, how the mitochondrial membrane potential is generated. Okay, inside every mitochondria there is a voltage of a difference, okay, like a tiny battery. So this is called what? The mitochondrial membrane potential. So it is the difference in electrical charge between the inside and outside of the membrane, that is what the mitochondrial membrane. Okay, so what happens in the mitochondrial membrane? An electrons move through the electron transport chain. Okay, that is what the through the ETC, we know that, right? They power pumps but the hydrogen ions into the intermembrane space, right? So this builds up what? The proton gradient, that is what the more and more the positive, that is what the protons, okay, they are going to what produced or accumulated in this the intermembrane space. And first thing, this is what the mitochondrial, okay, mitochondria, and we are now studying this portion of the mitochondria that includes what? The mitochondrial matrix, okay, intermembrane space of the mitochondria, and the cytoplasm. Okay, as we know that mitochondria, it is mainly made up of two membranes, okay, double membrane structure, right? So during this, what happens? This mainly occurs in the mitochondrial membrane potential occurs step by step. Okay, first thing what happens? The electron input in the citric acid cycle, what happens, or the Krebs cycle, it produces high energy electron, okay, electron carriers, that is what the NADH, okay, and FADH2, right? So here what happens? These molecules deliver their electrons to the ETC, okay, to the ETC located in the inner membrane, inner mitochondrial membrane, right? So this is what the electron input. Then next step, what happens? The electron transport chain, okay, what this ETC, okay, it will pass the electrons through a series of protein complexes, okay, through a series of protein complexes like protein complexes 1 to 4, right? They release what? Energy in a stepwise manner, okay. This energy is harnessed by certain complexes to pump protons across the membrane, right? This is what the ETC, it is electron transport chain. So next step, what happens? Proton pumping. So complexes mainly what? Mainly the first, okay, third and the fourth. As you can see that these are lined in the membrane, okay, intermembrane space. So this what happens? This will act as a proton pumps, okay, using the released energy to move the hydrogen ions from the mitochondrial matrix, okay, from this to intermembrane space, right? This active transport creates what? The charge imbalance and a proton gradient. Okay, what happens here? If it is happened like this, the concentration of a protons, okay, H plus ions, it will be lower in the mitochondrial matrix and it will get accumulated in the intermembrane space, right? So this creates what? The imbalance, okay, charge imbalance, and it forms what? The proton gradient, right? So next step what happens? Gradient formation. So the pumping of a protons establishes two interlinked, it forms what? The two interlinked gradients, that is what? The electrical gradient, okay, and the chemical gradient, right? So in the electrical gradient, the intermembrane space becomes positively charged relative to the matrix, right? So in the chemical gradient, that is what happens, the proton concentration becomes higher outside the inner membrane than the inside. It will be get accumulated all the protons here in this intermembrane space, okay. The proton concentration will be higher in this, okay, intermembrane space, and in the matrix the concentration is going to be decreased, right? So together these form, okay, these two forms the electrochemical gradient, also called the proton motive force, okay, that is simply we can call it as what? The PMF, okay. This is what occurs, okay. And next step what happens? Membrane potential generation. So the combined effect of a charge separation and proton concentration difference, okay, that creates what? The mitochondrial membrane potential. That is what? This one, okay. So this creates the mitochondrial membrane potential. Typically what happens? This potential ranges from minus, that is what? In the negative, negative inside, that is minus 152 minus 180 millivolts, 180 millivolts, okay. This is what? The potential range, okay, in the inside the mitochondrial, that is in the matrix, okay. Then what happens? ATP synthesis. So after that what happens? The ATP synthesis occurs, that is what? The stored energy in this gradient is utilized by the ATP synthase. Okay, and in the ATP synthesis what happens? The stored energy in this gradient is utilized by the ATP synthase enzyme, that is what? The complex 4, okay. This is what? The complex 4, it is going to utilize the gradients, okay, the stored energy, okay. And as proton flow back into the matrix through ATP synthase, okay, the enzyme uses this proton motive force to convert ADP to ATP. That is how the ATP synthesis is occurred, right? So this is the final step of oxidative phosphorylation, right, where electrical energy is mainly transformed into the chemical energy, right? This is what all about how mitochondrial membrane potential is going to be generated and how the ATP synthesis also occurs, okay. And next we'll see why this membrane potential is important. Why it is important? Why? Because it drives the ATP synthesis by the enzyme, that is what? The ATP synthase, that is complex 4, right? And also it maintains what? Okay, it maintains ion homeostasis, okay, and also it regulates the metabolite transport, as well as it indicates the mitochondrial health. It is a very important thing. Why? Because to occur all these things, the mitochondria, it should be healthy one, right? And also that is also measured, how we are going to see in the next slide. And also it is, why it is important means involved in the apoptosis, that is what? The programmed cell death, right? These are all what the important things or the concepts related to the mitochondrial membrane potential. So why we need to study this mitochondrial membrane potential, right? Next we'll see the measurement. So here what we are going to measure the how the mitochondrial health, or it is and in different ways we are going to measure the mitochondrial membrane potential. How? Because how means In the mitochondrial membrane potential, it is measured using the fluorescent dyes such as the JC-1, rhodamine 123 and TMRE or TMRM, right? Healthy mitochondria show a high membrane potential, okay, while the damaged or depolarized mitochondria, it shows what the lower potential, okay. This is how, by this, okay, we are going to analyze how we are going to analyze the healthy versus the non-healthy or mitochondria, okay. In the fluorescent dyes, what we are going to measure the mitochondrial membrane potential, okay. In case of the JC-1, what, and also in a TMRE, okay, that is what, a tetramethyl rhodamine ethyl ester, okay, are fluorescent dyes. These two are what, the fluorescent, these two are mainly the fluorescent dyes, okay, this one and this one. So these mainly used to assess the mitochondrial membrane potential by accumulating in a healthy polarized mitochondria, okay, and they mainly help in the distinguishing between the polarized, that is what, the polarized and the non-polarized, okay, polarized and the non-polarized, the mitochondrial states, right? In the JC-1, what happens, okay, in healthy mitochondria with a high membrane potential, JC-1, okay, molecule, it form what, the red fluorescent aggregates, okay. What it will do, it will form the red aggregates, okay, red form of a red fluorescent aggregates, that is what we can call it as what, the J aggregates, okay. And in case where it is also membrane potential decreases, when membrane potential decreases, JC-1 remains in its green fluorescent monomer form, right? And measurement, so how we are going to measure this? The red to green fluorescent ratio is used as an indicator of a mitochondrial health, okay. After we are going to see this, okay, J aggregates. So if we observed the color red to green, okay, that indicates what, the mitochondrial health. A decrease in this ratio signifies mitochondrial depolarization. Simple. If it is red in color, the mitochondrial healthy one, and if it is turns into green, okay, that J aggregates is converted into green color. So this says that the mitochondria is about to die, right? And uses. So uses of this JC-1, it is ideal for yes or no discrimination, okay, of a mitochondrial polarization. If the mitochondria is polarized or depolarized, it can be, by this we can going to analyze, and it provides a ratiometric measurement, making it more what, accurate for comparing samples, right? This is what all about the JC-1, okay. And coming to the rhodamine. So here what, sorry, coming to the TMRE, that is what, a tetramethyl rhodamine ethyl ester. So mechanism, TMRE is a cationic dye, okay, that accumulates in a healthy polarized mitochondria, okay. It will produce what, the orange fluorescence, okay, orange fluorescence when mitochondrial depolarization occurs, okay. TMRE is released due to loss of a membrane potential, resulting in a decreased the fluorescence, right? So how we are going to measure this? The intensity of orange fluorescence correlates directly with the mitochondrial membrane potential, right? A drop in a fluorescence indicates the loss of a polarization, okay. And main uses of this, the TMRE, it is mainly suitable for monitoring dynamic changes in a mitochondrial membrane potential in a live cells, okay. And that is how the very important thing is here, it is mainly used in the live cells and mainly commonly used in a flow cytometry and a fluorescence microscopy, right? These are what the main thing about these two. Okay, and main comparison between the JC-1 and the TMRE, that is what a fluorescence type. It mainly the JC-1, it produces what the red aggregates, right? So whereas in case of this, okay, it will produce what the orange aggregates. So read out what a green, red to this one, red to green ratio, and here in the it is what the fluorescence intensity, right? And application, where this JC-1, it is mainly applied in case of the ratiometric, and here it is non-ratiometric. And application, this is mainly used to tell the polarize yes or no checking. Okay, whereas in case of the TMRE, it is mainly used in the dynamic potential monitoring. Okay, and common techniques, it is mainly used in the microscopy and the plate readers, like that. Okay, whereas in case of the rhodamine, this TMRE, where it is mainly used in the dynamic potential flow cytometry, live cell imaging, like that. Okay, so scientists measures this voltage using the special fluorescent dyes like a JC-1 and a TMRE. So bright fluorescence is equal to healthy mitochondria. Let's see. So bright fluorescence, okay, here in this the JC-1, the red aggregates. Okay, you can see that in the picture, red aggregates, that shows what the high membrane potential. This mitochondria is a healthy one. When the membrane potential decreases, okay, low membrane potential, it leads to what the death of a mitochondria. Okay, this mainly suggests that the healthy mitochondrial status. Okay, and simple, we'll study what the key points to remember. So typical value of around this the mitochondrial matrix is about to minus 150 to minus 180 millivolts, and it is mainly generated by proton pumping via ETC complexes 1, 3 and 4. Okay, and it is mainly essential for the ATP generation, and loss of a membrane potential, mitochondrial membrane potential, is equal to the sign of a mitochondrial dysfunction or cell death, right? And summary, overall summary of this, we can say that the main feature and description. Okay, location, inner mitochondrial membrane, mainly this what the mitochondrial membrane potential occurs, and it is mainly generated by the electron transport chain. And function, it drives what the ATP synthesis, and typical potential minus 150 to minus 180 millivolts, and indicator mitochondrial activity and cell viability, right? This is what all about the membrane potential for mitochondria. So next time you hear about the powerhouse of the cell, remember, it's not just burning fuel, okay, it's running on electricity also, right? So if you like this video, please like, share, comment and subscribe my channel, The Igniting Curiosity in Life Science, for more and more biology videos. Okay, see you in the next video. Thank you.